Welcome AP Bio students. Today I'm recording on a Friday, so I am dressed in my Python gear. So let's try to keep up that Python spirit. The AP College Board gives a very structured class curriculum for teachers. And the first unit, if you take a look at the AP College course outline that is available to you on the AP College Board site, they want instructors to spend 5 class periods reviewing chemistry and what is called the macromolecules. And because I have some concerns as to how the whole year is going to go, and knowing how they altered the exam for last year's remote learning, I'm going to start the unit off a little bit differently with just a very basic review of what is called functional groups. And then also in this first lesson, what I call 1A, to start going over the macromolecules. So functional groups are, there are 6 of them, and we are going to learn them all for this first week. The only one I'm going to have you learn actually is or be able to identify is the hydroxyl group. And that one should be very familiar with you from basic chemistry. So functional groups are pieces that are somewhere along the line of a chain. So in chemistry or in biochemistry or your college credit chemistry class, if they're looking at a polymer or a chain, they sometimes will put for all of the carbons or all the other parts of the chain, they don't put down C's, they'll put down an R, the remaining of that polymer. And then if they put at the end or along the side another group, those are often called functional groups, and those are what can alter the compound or the molecule. So we have an amino group, carbonyl, carboxyl, hydroxyl, phosphate, and sulfylhydride. And so your phosphate and the sulfylhydride is our easiest of those 2 groups to be able to identify, and you'll see in a few minutes why. So here, this is an amine group. Here is the rest of my chain, where my R's would be. These would be carbons generally. And so it will be carbons. There'll be other things off of that. So here, the amino group has the N, which you'd be used to, and then our hydrogens attached to that. And as soon as you attach that amino group, it makes our substance a base. So again, very easy to identify. You're looking for an N. The carbonyl is a little bit more difficult to recognize. That has your carbon, a double bond with an oxygen. So this is going to be, and then here it has 2 other groups that are off of that. So here is a ketone, and they are hydrophilic. They like water. And an aldehyde is the exact same thing, except for, look at this. Instead of another carbon group coming off of this, we have a hydrogen. And it's easy for you to remember the difference between a ketone and an aldehyde because we have an H in aldehyde and an H off of our carbon group. So for a carbonyl, we are looking for a carbon with a double bond oxygen. Let's go back up for just a second and look at the amine group. The amine group, I'm looking for the N, the nitrogen. In the carbonyl, I'm looking for C for carbonyl and a double bond O, and we can have either a ketone or an aldehyde. And the ketones actually get to be important as far as anatomy is concerned because a buildup of these in the bloodstream can actually cause death. A person that has diabetic, or an awful lot of people died of ketoacidosis from the original Atkins diet, which has changed quite a bit. And again, C, double bond O, and an aldehyde would have the H. Now the carboxyl group is almost identical, C with a double bond O, but instead of having an H for an aldehyde, it has a hydroxyl group. And this is often with amino acids, and so this acts as an acid. That hydroxyl will turn it to acid. Remember, the N is for, in the amino group, is going to give us a base. Amino acids would have the carboxyl group. And the carboxyl is C double bond O with a hydroxyl attached to it. In this hydroxyl unit, although this here is a carboxyl, if you look at just the hydroxyl unit itself, they are, alcohols have it, they are polar, they are hydrophilic, And this is the one that I'm having you learn for this week, because some of our organelles, which you'll learn later on in the week, by adding hydroxyl group, it changes the substance so that it can be flushed out of the body. So that hydroxyl unit, as far as anatomy, is very important. And again, I told you that the phosphate was going to be pretty easy to identify. It has a big old P on it. So examples of with your phosphate groups are ATP, ADP, GTP, and these are probably not as familiar with you as I would like them to be, because these we would have emphasized when we learned the cellular respiration or the Krebs cycle, and we spent very little time on that because of remote learning. Here we have our good old S that identifies us for this group. It will be in some of your amino acids, and it is best known for stabilization. I will also tell you that the AP College Board will tell you that this is the least asked for functional group. It's very easy for you to be able to identify if it's in a content multiple choice question. But having said that, in the last exam that was due to remote, they did not ask any specific multiple choice questions. They were just generalized essays. So let's prepare as if you're going to be asked content and prep just in case. So the other thing you're going to need to know about is all of the different types of reactions: hydrolysis, dehydration, endergonic, exergonic, redox. And we're not going to worry about those at this point. I'm going to leave them in this PowerPoint, and it's going to go attached to this lesson 1A. But having said that, this is again things that I think, as I discuss as the course goes on, you're going to be able to follow. I will give you specific instructions as we are going along. This would be interesting to look at right now. Polymers are chains. You should have learned about that. Here I have that very famous hydroxyl group, and they're telling you if it's a dehydration, they're taking away water. Hydration, you'd be adding water. And again, I'm going to spend time as the course goes through, going through each specific reaction. So if I was a student right now, I'd be like, what is going to be on the first test? Do I have to learn this all on my own? No. So first of all, I've told you the only functional group you need to know is a hydroxyl unit. And now we begin that overview of the macromolecules. So these are our large molecules in the human body, and it's what all of biochemistry is based off of. So we have 4 large classes of very large molecules: lipids, carbohydrates, proteins, and nucleic acids. And for today, we're focusing in on lipids and carbohydrates. So lipids are better known as fats. But as you can see, there's actually 3 different categories of lipids. One of the important things to remember is that lipids are out of all the macromolecules, they're the only one that do not form those big chains. They're actually built by separate chains being put together. So lipids make sense, hydrophobic. They don't like water. Think of oil and water, trying to mix those, and the oil always goes to the top of that. And they are hydrophobic mostly because of what they have, what is called hydrocarbons. And hydrocarbons form nonpolar covalent bonds. And these are very important because remember, the plasma membrane is made up of phospholipids. So there's an awful lot of lipids within the body. So our 3 big classes of lipids are the fats, the phospholipids, and the steroids. So our fats by themselves are made up of glycerol and fatty acids, and they are, the glycerol is what we call a 3-carbon alcohol, so it has a hydroxyl unit attached to each of its carbons, and then a fatty acid has that carboxyl group. So remember, that's the C with the double bond O and a hydroxyl attached to that. And again, at this point, it is not important that you can recognize that they are 3-carbon alcohol. It's not important that you can recognize that carboxyl unit. You should be able to tell me, though, that there are 3 groups: fats, phospholipids, and steroids. Here's just an image of what that looks like. Here we have a fatty acid, and so I have our glycerol attached to that. And up here, remember that it said, if we're going to have, we'll have a glycerol and a fatty acid. And here we have to get them to join together. Water has to be removed. So then we have what is called ester linkages. And again, at this point, It's not important that you're going to know that. When we talk specifically about fats later on in the course, we're going to go back over how by removing that water, it has a very strange linkage called an ester linkage. Again, I have a feeling this is not going to come up in the course, and if it doesn't, if the AP College Board tells me that it won't end up on the exam, then we will review this as after the exam. And now, why do they separate from water? And this is a good one because remember, water is polar because it's covalently bonded, it pulls together like magnets. The two oxygens pull around that hydrogen, because water molecules form the hydrogen bonds with each other, and they actually push apart, water pushes apart the fats. So fats usually have 3 fatty acids attached to that glycerol. And if that's the case, we have a triglyceride. For example, here we have 1 fatty acid, and that's right here. And it's been attached to 1, 2, 3 fatty acid chains. So this is a triglyceride. And then we have 2 big categories of fats, saturated and unsaturated. So the unsaturated just means that there's at least 1 double bond, and that means that it is going to be more liquidy. The more unsaturated it is, the more liquid it is. So a saturated fat means there is not a single double bond in the go back up here. Saturated have the number of hydrogens and no double bonds. And here, this is confusing to the student because it is showing you that here, this is a double bond, a double bond, double bonds. So if I'm a student, I'm going, whoa, wait a minute, you just said there's no double bonds. What they're talking about is in this fatty acid chain right here that there's no double bonds. And so there's not ability to add anything anymore, and so it's solid at room temperature. In this one, where they do not show you, and they should show you, one of the worst pictures out of the book, in my opinion, is that there's at least 1 double bond available here. And here they have it. And because of that, at room temperature, it will be liquidy. And so our body needs fat. It is a high source of energy. And so our second big class of macromolecules that we're talking about today are the carbohydrates. And carbohydrates are what are broken down to make ATP eventually. Remember, they're broken down in glycolysis. So a very simple carbohydrate sugar would be a monosaccharide. So those are single sugars, for example. And then carbohydrate macromolecules are the ones that have many. So they could be a disaccharide or a polysaccharide. So to go over this a little bit deeper, for example, when we had glucose, which is a simple sugar, that was broken down outside of the mitochondria in glycolysis. And then the eventual byproduct of that, pyruvate, would enter into the mitochondria to make ATP. And so of the carbohydrate macromolecules, the polysaccharides are really made of many of those single chains put together. So fructose would be one of your very simple sugars. Glucose would be a simple sugar. And then our more concentrated carbohydrates, the things that we think of as carbs, starch, etc., would be so here it shows you that the more common glucose is the most common monosaccharide, and they're classified by that carbonyl group. And I have never known that to come up on one of the AP exams. So all you would need to know is that glucose is a simple sugar. And in fact, one of the labs that we do, the glucose, when we add it to a chemical called Benedict's and heat it, it's supposed to be, in students' minds, it should change color. But Benedict's it goes from blue to orange only when it gets reducing sugar or more complex sugar. So here it gives you some of those examples of the sugars. And a disaccharide is when we have monosaccharides put together through dehydration. So more than one chain. So here we are seeing glucose, we're having water removed, and we get maltose. Here glucose and fructose join together, and we're getting sucrose. So sucrose is a polysaccharide. And so they are well known for storage. Glycogen is converted from glucose into a storable form in the liver. Starch is another form of a carbohydrate that is stored. oftentimes in plants. And the polysaccharide cellulose can't be digested by people, and that's the main one for plants. And enzymes can break these down, but not cellulose.